Thank you for inviting me to speak at the Global Health Security Innovation Week. My name is Laura Newcomb, and I'm a biology professor at California State University in San Bernardino. My talk today is entitled Zoonosis Happens. Zoonotic diseases are spread between animals and people. This talk will focus on viruses that are emerging zoonotic threats to global health and security. I'll specifically talk about coronavirus and influenza virus, two that you may be familiar with. However, there are many, many more viruses that have zoonotic potential. However, I would be remiss if I did not tell you that most viruses do not threaten our health. Indeed, viruses are important and essential life entities. For example, bacteriophage, or viruses that infect bacteria, are responsible for regulating bacterial populations, and therefore they're essential for global nutrient cycling, and they're essential for maintaining the health of your microbiome populations. So very important jobs. In addition, viruses have had very important roles in our evolution, particularly retroviruses, because they must integrate their genome as part of their replication cycle. And so if they integrate their genome into a germ cell, that can be passed on. And over time, this what's now termed endogenous retrovirus can be repurposed for host function. And a good example of that is the evolution of the placental syncytial genes from retroviral fusion proteins. So although only a small fraction of viruses cause pathogenesis, unfortunately, as we've learned firsthand, that small fraction can really wreak havoc on global health security. So as of August 31st, over 4.5 million people worldwide have died due to COVID-19. Over 640,000 of those in the United States. So why does zoonosis happen? Well, one reason is because all life is made of the same molecules. We are truly connected. And therefore, some viruses can infect multiple animal hosts. So here is a depiction of some of the viruses that are known to infect both animals and humans. There are others. I'm going to focus on coronavirus and influenza virus, which is part of the Orthomyxoviiridae family. So both of these viruses infect many different animal hosts. This is a depiction of the different animal hosts that can be infected with influenza A virus, with wild aquatic birds being the reservoir. And then this is a depiction of the zoonotic coronaviruses. And again, you can see just from this depiction that there are many different animal hosts that coronavirus can infect, some of them being domestic and some being wild. So in both cases, these viruses can infect our domestic animals and wild animals. We have close contact with animals, and so we have close contact with these animal viruses. So for example, our livestock that even if we don't interact with ourselves, we typically consume their products. We often have pets. Further, we know viruses can be airborne, and some viruses, like for example, West Nile virus, are transmitted through insect vectors. As we encroach more on wild habitats, it is likely we could encounter additional animal viruses that we have yet to discover. And so viruses evolve quickly and they're subject to selection. So therefore, as we are exposed to those viruses, we could evolve a virus that has easy zoonotic abilities. And so viruses make mistakes during genome replication. These changes are referred to as antigenic drift and are thought to be, small changes, and they're due to errors during replication. So viruses have relatively small genomes, but they make relatively large quantities of progeny, and therefore they're replicating their genome to an abundant level. And in some viruses, their replication is error prone. And so in some viruses, it's estimated that each genome copy has at least one nucleotide change, even in a single infection cycle. And so within an individual, this can be termed quasi-species, all these different variants from virus infection. Viruses can also change through other mechanisms, such as reassortment and recombination. And this causes a larger change and is considered antigenic shift. And so, for example, with influenza virus, which is a segmented genome virus, if two viruses infect the same host cell, these segments during replication can reassort in many different combinations. And so you have lots of different progeny, some with new combinations of surface proteins that has not been observed before. Now, coronavirus is not a segmented genome virus. It has a long RNA segment, but it's a very long RNA genome. It's actually more than double the size of all of the influenza segments together. And the polymerase tends to fall off and reanneal during replication and RNA synthesis. And so coronaviruses have a high propensity for recombination because the polymerase will fall off of its initial template and then recombine with other similar coronavirus templates that are replicating in the same host cell. And this will result in progeny that have new combinations of the coronaviral genome, resulting in new combinations of the different spike proteins. However, both of these molecular mechanisms of change in viruses can result in zoonosis and even pandemics. And so we know this from influenza virus, where we've had more experience dealing with pandemics. So influenza was responsible for pandemics in 1918, 1957, 1968, and most recently in 2009. And of course, influenza still remains a concern for an emerging pandemic. But the 1918 influenza virus was found to be a completely avian influenza virus, so no reassortment, likely changes due to antigenic drift that caused the virus to gain properties to infect humans. The other pandemics were due to reassortment events, typically between an avian influenza and human influenza. But most recently, this was a reassortment. The 2009 novel influenza was this reassortment between human, avian, and two different swine influenzas. So for coronavirus, we know that, again, that both of these mechanisms can result in a change, for example, in this depiction of a surface protein that does not interact with the human receptor very well, but either by drift or shift gains the molecular characteristics necessary to interact with the human receptor. And so we know for SARS, there was an intermediary that the virus went through, and the SARS from 2002, 2003. And similarly for MERS, an intermediary was identified. For SARS-CoV-2, the cause of COVID-19, an intermediary animal has not yet been identified. And further, it could be that there is no intermediary animal that was necessary for this zoonotic event to happen, because it was known even in 2015, that by sampling coronaviruses, there were viruses circulating that encoded spike variants that would be compatible with interaction with the human ACE2 receptor. In addition, in 2018, it was shown that there were individuals which were living in villages near bat populations that showed positive reactivity with SARS-related coronaviruses, so bat SARS-related coronaviruses. And so these are 6 individuals here that were showing cross-reactivity. Over here, we have controls of negative and 1 positive individual who had suffered SARS in 2002, that sample. And so it is possible that there was no intermediary for the SARS-CoV-2, similar to the 1918 influenza pandemic, which went straight from the reservoir birds to humans. So what are the changes to the S protein in SARS-CoV-2 that allow it to enter into human cells readily? And so if we look here, this is the genome of a coronavirus with the spike protein, which is magnified here. And the SARS-CoV-2 has needed changes in the receptor binding domain that allow it to interact with ACE2. Some of these are found in bat SARS related viruses. And initially, early suggestions were that maybe pangolin was an intermediary. Some of the changes were found in pangolin. But also these changes are, small and could occur just through replication error. Another feature is this addition of a polybasic cleavage site. Now, while this sequence isn't in these closely related coronaviruses, this sequence is found in other coronaviruses, so it could have been obtained through a recombination event. But also, it's again a small sequence with repeated amino acids. So these arginines are basic amino acids. And so that could also have easily evolved during insertion errors during replication. And so this actually happens with influenza virus. It's been demonstrated that there is a predisposition to obtain these polybasic cleavage site within the surface protein of the virus that's important for entry and which needs to be cleaved for entry into the cell. And so you'll notice here that the basic amino acids are encoded by codons that are repeats of adenine. And so that can easily be an insertion error as a polymerase jumps off and reanneals to the template. And of course, this polybasic cleavage site does increase pathogenicity, as is depicted here, because it's allowing the virus to be more readily taken into the cell. And so for influenza, we know other amino acids that are important for the HA surface protein to make it compatible with replication in human cells. And so some of these sites are known and what allows the viral HA to bind to the human receptor. And over here we have other changes that are known to increase viral transmission in a ferret model. So we know some of the changes for influenza HA to look for potential emergence of novel zoonotic influenzas. In addition, we know other characteristics for influenza that promote infection in human cells. And so here we have 3 residues that are known to be important for efficient replication in human cells. And we know which amino acids are preferred for that to promote replication in human cells. And this is within the influenza polymerase basic subunit, part of the replicase enzyme of the virus. And indeed, looking at surveying some of the circulating avian H5N1 influenzas, some of them already harbor these changes that would improve influenza replication in human cells. So we know a lot about influenza because the World Health Organization has been surveying and responding to influenza at the global scale since 1952. And this is the kind of thing we need to do to be better prepared to both predict the next pandemic and stop the next pandemic from happening. So we need to be aware of all potential zoonotic viruses. We need to understand the molecular changes that are required for those viruses to readily infect and transmit in among humans. And we also need to be conscientious of where we are interacting with animals and how humans and animals interact. And so there is an initiative that started in 2016 called the Global Virome Project, which is a collaborative initiative basically to discover potential zoonotic viruses, work to build capacity to understand and prevent any spillover events from happening, and then foster solutions if these events do happen, and again, prevent these events as well. And so I hope that I've convinced you that zoonosis happens, and it can happen relatively easily. And this is because all life consists of the same molecules, and therefore some viruses can infect many different animals because we really are very similar, especially at the molecular level. We live and interact with animals, and therefore we come into contact with animal viruses. And viruses change constantly. Every replication cycle, they are changing, and those changes are selected for. So viruses replicate rapidly, produce lots of progeny. The genome changes are subject to selection, even within an individual. And so you can understand how easily that evolution can happen with viruses. And even small changes can have big functional changes. So we saw just a few repeats of nucleotides being added could add basic residues that can provide a new cleavage site so that the virus gains entry into human cells more readily and expands its host range. So what we need to do to be prepared for these potential pathogenic viruses is we need to support science, sampling, and sequencing. And so we need to support and fund the science to understand the molecular properties that allow animal viruses to efficiently infect and transmit among humans. So what kind of surface protein requirements, what other species-specific determinants are important. We need to do sampling to understand what viruses are circulating and where are these viruses present and where are humans interacting with animals that contain these viruses. And then, of course, sequencing. Once we've identified changes that are potentially detrimental to humans, we want to identify those changes and work to inhibit any emergence of a new human pathogen. And so with that, I want to thank you for listening. I've included my email if you want to contact me. And my lab at Cal State is primarily focused on identifying novel antiviral targets to counter emerging influenza A viruses. So thank you for your time.